So welcome everybody. My name is Kelly Street, and this is the Trajectory Inference Across Conditions workshop. Okay. So like I said, this is the Trajectory Inference Across Conditions workshop, differential expression and differential progression, which is kind of a term that we made up. So we'll be sure to explain that very thoroughly. My name is Kelly Street, and I am happy to be joined by my fantastic collaborators, Koen van den Berge and Hector Roux de Bezieux, who's joining us despite some last minute changes of plans. But both of these guys are excellent collaborators, so you should all take any chance you have to talk to or work with them. And then down at the bottom, we have a link to our GitHub page, which has the full workflow that we're going to be presenting today. I believe Koen also posted in the chat a link to the instance of our workshop if you want to play with it yourself. So I'm going to start by presenting the data set that we're going to be working with today, which is just one example of the type of data set that you might be working with and that you might apply these methods to. But it's a very straightforward example, and it fits very well with what we want to talk about. So this is sort of a simplified version of this experiment where we're studying EMT, epithelial to mesenchymal transition. So they take these epithelial cells and plate them and let them grow for a certain amount of time. And as they divide and differentiate, they turn into mesenchymal cells. And then from the second plate, they collected both an inner and an outer sample. The inner sample tended to be more epithelial cells. The outer tended to be more mesenchymal. The original paper, by the way, I've listed at the bottom, a pooled single-cell genetic screen identifies regulatory checkpoints in the continuum of epithelial to mesenchymal transition, is a very well-written paper and definitely worth reading. And they present their own methods for dealing with these sorts of analyses. What we're interested in is what to do when you have this sort of experimental design where you're basically doing that twice. We've got this single continuum of cells from epithelial to mesenchymal, but we've got it under two different conditions. We've got an untreated or mock group, and we've got another group that's been treated with some sort of drug, in this case, TGF-beta. And so we're interested in comparing the development under these two conditions. So this is a sort of the upstream workflow. We're gonna gloss over this for the most part because this is not the main focus of our workshop, but all of these steps are important and worth considering and, you know, very much not like decided. So these are choices that we made along the way to get to the part that we wanted to talk about, but each of these could individually be debated for a long time. So we're using Seurat for both normalization and batch integration. So I showed the experimental design a minute ago. We had to collect, or they, not we, had to collect the cells from the two different conditions separately. And so one of the first things we have to do after normalization is to integrate those two data sets so that they somehow match or, you know, can be represented in the same space. And then trajectory inference, we want to identify that primary axis of epithelial to mesenchymal transition. And we're going to use Slingshot for that because I developed Slingshot, so it would be weird if I used anything else. So one of the first steps in our workflow that we're going to talk about more in detail in a minute is differential topology. And by this, we mean looking for differences in the large structure of the conditions. And so you can see in the top left here, panel A, we've got a single trajectory and two conditions that are sort of evenly mixed throughout. But we could just as easily have a situation like panel B, where the two conditions diverge and they end up in different places. And so if something like that happens, it might not be appropriate to fit a single trajectory to both conditions. This might not be, it might not be appropriate to fit, say, a branching trajectory because the fact that, you know, an early cell is condition B means that it's already determined it will end up in this branch, not in this branch. And so it might not be appropriate to model that as a branching trajectory, but rather two separate trajectories that happen to overlap. Another problem that we're going to tackle is differential expression. And this is going to be in the context of continuous pseudotime. And we have a package for this called TradeSeq that we think is very well suited to handle this differential condition problem as well. And so this will allow us to answer questions like, are these genes exhibiting dynamic expression over pseudotime in either condition? And are they exhibiting different patterns over pseudotime between the two conditions? And so these are very similar to the types of questions that we used to ask of branching trajectories, where we could say, is a gene following the same pattern in this branch versus that branch? Now we're just talking about, is it the same pattern between conditions? And so this is, I think, a very good fit for the types of models that we had already developed with TradeSeq. And so with that, I'm going to switch over. I'm going to point out our GitHub page if anybody wants to play around with anything there. But for the most part, the Zoom stuff keeps getting in the way, sorry. I'm just going to present from our website, which is kstreet13.github.io/bioc2020 trajectories. And this is the website generated automatically by our repository. One more time, I'm going to point you at the original paper, which, if you're interested in these sorts of methods, is a very interesting paper, very much worth reading. They present sort of the monocle approach to these questions. And yeah, we're basically just showing that, you know, we used our own methodology, but we have similar results to theirs. So jumping in, I'm going to skip over the introductory stuff because all of that stuff that I just talked about was introduction. This is our upstream workflow, which, like I said, we're not going to talk about a whole lot, but here's all of the code that we used to run SC transform normalization with Seurat, which is down here, to do the latest and greatest Seurat integration. And then we're converting between Seurat objects and single cell experiment objects using these as.single-cell-experiment and as.Seurat functions. So we're not actually running this code in our workshop. We're actually cheating and just importing the data afterwards. But all of this only took less than 10 minutes to run on my laptop. It's actually surprisingly fast. So feel free to, you know, ask questions, comment on any of that. I would say that's fair game. But we didn't include the actual running of that code because, like I said, we're focused on downstream. So then the first step, like I said, is this differential topology question. Do we see between the two conditions very divergent sorts of topologies where, you know, they might be completely distinct? They might be two lineages that are completely unconnected, or they could have, you know, one connecting point and then branch off, or be perfectly overlapping. And so what we see here in sort of in our UMAP representation of the data is that we do have pretty good overlap between our two conditions. The Mach and the TGF beta, in red and black on the left here, both are very well represented throughout this lineage. And then I mentioned at the beginning, we've got two different areas of the plate from which cells were collected. There's inner and outer. And we can kind of see that there's, you know, more inner cells towards the left, more outer cells towards the right. And so that gives us an idea of the orientation of this trajectory. That probably indicates that the left side is our early pseudotime and the right side is our late pseudotime. And so this is what we're going to be talking about when we talk about differential topology. We're looking basically at neighborhoods around each cell and asking, does the composition of cells in that neighborhood match the overall composition of the conditions? So we've got a more or less even split between our two conditions. So within each local neighborhood around a given cell, we want to see a more or less even divide between the two conditions. If you're familiar with KBET or KBET, which is a metric for evaluating normalization methods. That's a very similar sort of metric. We do some smoothing. I should point out the function that we use here is imbalance score. K gives us the neighborhood size around each point, and smooth is a smoothing parameter to say how much we should smooth these, because you can see that there has been some smoothing in this plot down here. And what we see is that, you know, there are a few sort of small neighborhoods where there's imbalance, but for the most part, we see a lot of purple here. It seems like we've got pretty good mix between the two conditions, and that's what we saw earlier when we were looking at the red and black conditions plot. And so with that information, we feel pretty comfortable fitting a single trajectory. It doesn't seem like there's any obvious distinction where like being in the normal versus the treated group determines your outcome. It seems like both the start and end position is more or less the same, regardless of which treatment condition you're coming from. So that gets us into trajectory inference, which is going to be a pretty straightforward application of Slingshot. The only interesting thing here that I want to point out is that Slingshot works on a minimum spanning tree of clusters. And so oftentimes we would include a clustering step at this point, where we just do something very simple, maybe like k-means with a k of 3. But in this case, we don't actually have to do that because we've got this inner and outer information already. And we know ahead of time that the inner group of cells should be earlier in pseudotime, and the outer group should be later in pseudotime. So what we did here is we treated those as our clusters, and we just made a minimum spanning tree on those two clusters, which is of course a straight line, which we can supervise and say, okay, we know that the inner group should be our starting cluster, and that ensures that we have the correct orientation to our eventual trajectory, which is shown here. And then just looking at the distributions of cells collected from the inner and outer portions of the plates, we do see that the inner cells are sort of skewed towards the beginning, and the outer cells are sort of skewed towards the end. And so we've already covered a lot. I think this was one of the places where we wanted to take a break and see if anybody had any questions from the polls.
Yeah, there's a few people already asking questions about later steps, and I suggest we do those when we're at those respective steps.
Okay.
There's a question on, do you have a suggested dimensionality reduction technique for trajectories? Diffusion map versus UMAP, etc.
I mean, I can say that I usually try to use PCA first if I can, but that's not always possible because sometimes you've got more complex trajectories. But UMAP I've had generally pretty good results with. Diffusion maps were the other one that was mentioned. Mm-hmm. Sometimes, I mean, you know, maybe I'm just not using them correctly, but I find little like stringy pieces, highly linear branches off of the main cluster of cells. So I don't know. I think it's more of an empirical thing. I can say that I've had better luck using UMAP than anything else.
I agree. I think a general recommendation is definitely to try and use something that tries to keep the global structure of your data, fitting trajectories, and probably not something like TSNE, which favors local neighborhoods. We have another question. After integration, do you use the batch corrected expression values or the original raw counts with a batch factor? I'm asking because the batch corrected values from MNN integration should generally not be used for quantitative analysis. So I guess that this is already kind of pointing at the later differential expression analysis. And there we are not using the batch corrected values for differential expression because we've also seen that these are not recommended to be used. But we do the batch correction or the integration for the trajectory inference and to get the trajectory right. So there's one more question. When assigning clusters using known data parameters, are you setting the centroids for those data?
Are we setting it? We're not setting the centroids. We're using the centroids basically. But it doesn't matter in this case because there's only two, right? So the minimum spanning tree is going to be a line between these 2 points. We're really just using it because A, we know that we only want a single trajectory. So it's fine if our minimum spanning tree is just a line. And B, the inner versus outer gives us a way to order or set the orientation of that line because we want to make sure that we don't end up with a line that's going backwards.
Okay, I think we're good to proceed.
So that gets us into sort of the first test, like actual formal test that we run. And this is what we're calling differential progression. So the question here is, if we compare our two conditions along this now common trajectory framework that we've established, do we see any differences between the condition? We wanted something that sounded like differential expression for this, so we went with differential progression. Just looking at this density estimate of the two conditions, you can see that there definitely is some sort of difference between them. We see this interesting sort of trimodal structure in the untreated group, and then the treated group is sort of missing, or certainly has a lot fewer of that early cell type. It seems like they differentiated a lot faster or a lot more. And you would expect, just based on this plot, that hopefully we do see some sort of differences here. And essentially all we're doing is comparing these two distributions. There are a lot of different ways that cells could show different behavior along a trajectory. And so we want it to be as general as possible. It might be the case that one condition was more bimodal. So it's either all at the beginning, didn't differentiate at all, or all at the end, fully differentiated, whereas another one is sort of more uniform in the middle. So things like a t-test or anything else that is testing some sort of centrality parameter, we didn't think would be super appropriate. And so what we went with was a Kolmogorov-Smirnov test, which is a test to compare these distributions and ask, are these coming from the same CDF? And unfortunately, this output, this block just decided that it wasn't going to output anything in our final version. Every version up until our final version had the output. So I'm just going to switch over real quick to a version that I just recompiled on my computer and show you that we do get that good old classic 2.2 times 10 to the negative 16th p-value here. These are very much different distributions that we're seeing. And that's not super surprising. That's really just sort of a sanity check. But I think this is a good general framework for comparing conditions along a single trajectory. However, it's kind of crude. It's really just asking one single question, and it doesn't have a lot of fine level detail. And so if you want to ask some more detailed or potentially more interesting questions, that's where we're going to start looking at things at the gene level and asking about differential expression. And so this is a pretty, I think, good application of the TradeSeq package, which all three of us worked on developing, which is made for comparing different branches or assessing differential expression along a single branch of a trajectory. What we're going to be doing is fitting negative binomial general additive models, or NBGAM models, NBGAMs, which is going to give us, for each gene along a trajectory, some sort of curve that represents the average gene expression. And these curves are based on smoothing splines. And so the first thing that we have to do is determine a number of knots, which is essentially a number of degrees of freedom for those smoothing splines, so how adaptive we want them to be. And so for that purpose, we developed the evaluate K method, which lets you put in a range of possible knots, and it'll print out some summary statistics that you can use to hopefully determine an appropriate number. And so this can be a little bit computationally burdensome, which is why we've only tested 5 values here. If you're going any lower than 3, then maybe a quadratic model would be better for you. There's not a lot of flexibility at lower values, but we could have certainly extended a little bit further off the end, higher than 7, if we had sort of more computational resources to work with. But this is the idea, that it's going to show some summary statistics of how well the trade-seq model fits for various numbers of knots. And what we're looking at here is just a few different summaries. I'm going to focus on the average AIC, but that's the same as the relative AIC, where it's just relative to the highest value. AIC, Akaiki's information criteria is hopefully familiar to some of you. But essentially, we're going to see better and better fits, which means lower and lower values here as we get more knots. It's not surprising, right? The more knots you have, the lower the residual sum of squares, the better fit you're going to get. But at a certain point, we start to see sort of diminishing returns on increasing the number of knots. And so you can think of this similar to a scree plot, where you're trying to pick the number of principal components. We're sort of looking for an elbow point here. And I think if this plot wasn't so vertically stretched, you would sort of see it around 5. And like I said, when we're actually running our analysis, we might go out to 10 or 15 or a higher number of knots. So hopefully we could really see that this pattern continues after 5. But 5 is the number of knots that we decided to go with for this analysis. You could certainly make a case for several other numbers, but I just wanted to sort of describe our thinking about how to go about that. So once we've chosen a number of knots, we're going to use the fitGam function to actually fit our model to all of the genes. And I'm going to make special note of the conditions argument. So this is not available in the standard version of TradeSeq, the version that's currently on Bioconductor, but it is on our GitHub under the conditions branch. So right now, this is still very much in development. These are fairly new methods for us. And so you have to install via GitHub, and specifically the conditions branch, if you want to access this functionality. But otherwise, all we're doing is, like I said before, we're fitting a NB-GAM model. We have the original counts, which somebody asked about. We have the pseudotime. We have the weights, which are not particularly interesting in this case because everything is assigned to this only single trajectory that there is. And then we have the conditions and the number of knots. And this is going to fit our model, which we will then use to assess dynamic expression and differential expression between conditions. So that first question, which is again a classic TradeSeq sort of question, is do we see genes that have dynamic expression patterns? We would certainly expect to see that because we're seeing different cell types. So as cells are changing types, we would certainly expect to see some genes changing expression. We're going to use the association test for that. So this is testing for any association between gene expression and pseudotime. It doesn't have to be monotonic. It could be, quadratic or any other pattern that our smoothing splines are capable of picking up. And we're setting a cutoff on the log2 fold change so that we sort of only pick up the bigger changes. And this is not looking specifically at the conditions, although we are fitting it separately. No, I'm sorry, we're fitting it once, but then adjusting the p-values within condition. And so we've got separate sort of dynamic genes within each condition. And what we see is that we get quite a few, almost 2,400 dynamic genes or genes that are associated with pseudotime in the mock condition, and a little less than half as many in the TGF-beta condition. And then this is an upset plot that shows the overlap between those two sets of genes. And we see that there's very strong overlap. Almost all of the genes that were dynamic in the treatment condition were also dynamic in the mock condition. And then there's a bunch of extra ones in that mock condition. We can also visualize the pattern of gene expression using a heat map. And so here we're looking at gene expression over pseudotime. And these are the genes from the mock condition. So they show some sort of dynamic expression in that condition. And we see a few different sort of broad patterns. There are genes that are highly expressed early in pseudotime and then shut off. There are genes that are off early and then turn on late. And then various patterns of, off, then on, then off again at different points along the lineage, particularly in this middle section and this sort of 3/4 section. And if you wanted to do, I should also mention, if you wanted to do clustering on expression patterns, that's another functionality that we don't really explore in this vignette, but that TradeSeq does have. So if you want to cluster, we offer some sort of methods for clustering on the coefficients of the smoothing splines, which can be an efficient way to cluster based on expression patterns. Okay. And then we did some gene set enrichment analysis on those genes, which again is not the main focus here, so I'm going to brush over it a little bit, but we do see epidermis development, epithelial cell differentiation, a lot of things that we would expect to see from this sort of experiment. And then the final question that we're interested in answering is, do we see different patterns between the two conditions? And this is really the crux of the workshop. Do we see, genes that are behaving differently despite there being a single trajectory? What are the more subtle differences that we can identify between the two conditions? So just sort of as an exploratory analysis, here's CDH1, which is mentioned in the paper as sort of being similar between the two conditions. And it's broadly the same sort of pattern, up and then down. There's a little bit of a difference early on. CRB3 is the same thing, high to low, pretty similar between conditions. The original paper also mentions a few other genes that are very different between conditions. FN1 we see is almost completely off in the untreated and shows very high and increasing expression in the treated. Similarly, but less obvious, CDH2 is almost entirely off in the untreated and shows some fairly robust expression in the untreated. So that's all exploratory and based on the results from the original paper. For our analysis, we're going to use this new condition test, which again is only available in this latest version of TradeSeq that I mentioned, with a fold change parameter to set, or a fold change requirement. And this is going to do something very similar to the pattern test, for those of you that are familiar with TradeSeq's pattern test, where we're comparing the curves between the two conditions, and we're asking, do these have the same pattern of expression? And so smoothing splines are based on coefficients, which I mentioned earlier. We're basically testing, are those coefficients the same between the two conditions? And so this is ultimately a walled test under the hood. Sorry, I should mention, we're running that test and then doing a p-value adjustment, and we've got almost 2,000 differentially expressed genes, or differential in terms of showing different patterns between the two conditions. So our most significant gene is plotted here, and you can see it's very strongly different between the two. It's not surprising. This is sort of one of those that you would certainly hope you would pick up. Similarly, our least significant gene, pretty similar pattern. It's mostly off throughout both conditions. And so just as a sanity check, like, this makes sense. This is probably not differential expression. Again, we can do a heat map to look at the different patterns. And this time we're comparing the two conditions. So we include both the TGF-beta and the mock conditions. And we're ordering by this hierarchical clustering of the TGF-beta patterns. And before I, without talking about any of the specifics, you can clearly see that that pattern is very strongly degraded in the mock. And that's good. That's what we were hoping to see, because we're looking specifically here for genes that show different patterns between the two conditions. So if these two heat maps were identical, that would be worrying, because that would mean we were picking up genes that had the same pattern in the two conditions while looking for genes that have different patterns. And so we see the same sorts of things that we saw before, genes that are highly expressed and then turn off, genes that are off and then turn on, and intermediate genes that are on at some point along the way. And for almost all of them, you can sort of see similar patterns in the mock, but it's very much a muddled version of that pattern. It's definitely not as clear. There's some streaks of blue throughout that are just genes that are off the whole way. And so this is fairly reassuring that we are in fact picking up genes that show different patterns between the two conditions. And then just as a last step, we once again ran some gene set enrichment analysis and found some markers for locomotion and cell motility, things that we think are related to the treatment and similar to the types of conclusions drawn from the original paper. And so with that, I'll again point you to our website or our GitHub repository, and we will open it up to questions. Thank you.
Thanks, Kelly. So there's a few more questions. First one is, when combining data sets, how do you generally infer that a trajectory is real or accurate? A trajectory could be drawn between unrelated cells simply because they are in the same data set. And I would say to that, a lot of it depends on the experiment itself. Do you expect a trajectory biologically or not? If you're looking at, for example, the data set we've looked at, we really expect a kind of dynamic change when epithelial cells develop into mesenchymal cells, and you could expect the same thing for example, stem cells developing into mature cells. But if you would say, take a sample of a liver tissue, I wouldn't, even try to fit a trajectory, because there you would rather expect kind of discrete cell types, discrete cell populations.
I would also add, just as a minor point, that this is actually something I've been thinking about a little bit recently, thanks to Aaron Lunn, in the context of fitting trajectories in Slingshot. We have been working on sort of better ways to separate distinct populations. And that's always sort of something that I've been aware of. And so I think we've got a rule of thumb built in to Slingshot for how separated do clusters have to be before we say that they're not on the same trajectory. It works in some cases pretty well, in some cases not as well as we would hope, but there's at least some consideration of that. And that's a parameter that you can play with if you want to try to make sure that things that don't belong on the trajectory aren't accidentally included.
Hector, if you have anything to add, or if you want to share some views, feel free.
No, I agree that the integration can sometimes be tricky. If you have multiple data sets, sometimes you can see the trajectory in every data set individually before merging them. And that's a pretty good indication that something is really popping up.
Okay. The next question is, how do you prioritize genes for input to the fit-gam function, most variable? I would say that I would recommend some kind of low-level filtering where you would maybe just remove the genes that have low expression in general. we do realize that fit-gam function can be computationally intensive if you need to estimate a lot of parameters and if you have a lot of genes. If you want to filter genes, then filtering the most variable I think may not be the best idea because it may not be an independent filtering criterion. So in that case, I would rather try to maybe filter on the mean rather than the variance, because I would think that filtering on the variance might inflate false positives. So there's a paper on that from Wolfgang Huber's group I can share in the chat. So next question, can you explain more about the directionality? Would you get a different trajectory with different supervised directional assignments? Could the UMAP plot have arrows?
The directionality doesn't actually, it doesn't actually affect the final trajectory. It's just so that we know it's going left to right rather than right to left. In this case, since it's just a single lineage, we're basically just fitting a principal curve, which is a nonlinear generalization of PC1, basically. It's a line that tries to fit the middle of the data. And so much like doing a principal components analysis, the positive negative directions on that are somewhat arbitrary, but it would be the same regardless of directionality. It would be the same line.
And the input to the GAM model as well. If you were to switch pseudotimes just by putting a negative in front, you would get exactly the same results.
So next question, can you please explain the concept of knots in reference to pseudotime trajectory? Are these just multiple branching points in the pseudotime trajectory? So the knots are kind of, do not really relate to the trajectory that's been inferred, but more relate to how complex we expect the gene expression patterns to be over pseudotime. So if you have more knots, you will be able to fit more complex patterns, while if you have fewer knots, you will not be able to pick them up. For example, if genes suddenly increase quite drastically in expression, you will need a few knots to really pick up that peak and then go back down again. So the knots really relate to how complex the gene expression patterns are in the data sets. And there is no immediate link with the number of branching points in the pseudotime trajectory. So next question is it possible to include a temporal dimension, for example, treated, untreated, at different time points? If yes, would you specify them in some way or leave the pseudotime or trajectories take care of it in an unsupervised fashion? I'm not sure if I'm following the question.
It sounds like they're asking how would we handle multiple time points, which is kind of what the inner and outer is. I would think of the way that we handle inner and outer as being analogous to how we would handle time points, because usually, if you're collecting single cells at discrete time points, there will be some overlap in the types of cells that you get at each time point. Some, they're not differentiating synchronously, so you'll get sort of different distributions of cell types at each time point. And so you can treat the time point as a useful way of supervising the trajectory inference, but I would use the pseudotime rather than directly incorporating those time points as a variable.
Next question, can your differential expression methods be extended to multiple replicates for mock and TGFB? So this is a good question, and it's something we will be looking into. For now, we allow you to add fixed effects. So we allow you to add a replicate effect as a fixed effect to the model, which is okay if your treatment effect is, if you need to do differential expression within sample, then a fixed effect will be appropriate. If you're comparing, for example, healthy versus diseased patient samples, then you may want to consider something more complicated than simply adding a fixed sample effect. But we have not yet implemented that in TradeSeq. Hector, if analyzing multiple trajectories, do you need to evaluate K differently? So we do recommend you to evaluate the number of knots you want to use for each trajectory separately, because the gene expression patterns might be pretty different between trajectories, although we have found that the exact number of K doesn't make a huge difference. And typically the most data sets are between 4 and 8 knots. That seems to be like where most trajectories fit well.
I would add on that, because I think there might be a confusion between trajectory and lineages. What we call a trajectory is the cells start from one single common ancestor and then can go into multiple branches, which we call lineages. So if you have one trajectory like that, like kind of a tree structure, then you would just need to run evaluate K once. And then if you have multiple trajectory, which means multiple starting point, then we would recommend you run evaluate K for every starting point independently.
Next question, are the heat maps of genes DE between conditions independently Roux scaled? So yes, they are. Or on the same color scale. The former could make it more difficult to compare patterns that are similar, with one showing weaker variation. So they are scaled. So we scale each gene to have a mean 0 and unit variance. But they are also indeed separately scaled for each condition to make it more clear. Yeah, that's what we're doing. I'm not sure if we check the heat maps without the scaling, I should say.
Yeah, I think I can see what they're saying, where if you had similar patterns, but it was sort of a shift between the conditions, that might not show up in the heat map, but that would still be something that we picked up with the test.
I think there's a general problem. It's idea with these visualizations that if you don't scale, you might just get a completely blue heat map because of some outliers, and if you do scale, there's some issues. So it's really a visualization thing, but it's a good question and something to consider. The next question is, what if there are some trajectories which are highly dynamic and some that are less dynamic but still changing than others? Is there a point where using one value of K is suboptimal for capturing a reasonable fraction of the data? Maybe this doesn't happen biologically though. So I would answer to that we can easily handle that, because if some lineages are dynamic and others aren't, then the way the smoothing works is that there's shrinkage, and the optimal shrinkage is identified through a cross-validation approach. So if it's useful to fit a complex pattern for one lineage, then we will move to a higher K value when we run evaluate K, but that doesn't mean that the functions become overly wiggly for another lineage, because we have the shrinkage.
I think a second point is that the most dynamic lineages will tend to be longer because they will impact more the reduced dimension. And so this is going to also be reflected in TradeSeq in that regard.
So there's the last question is, I'm curious if this is suitable for solid tissues, given we have different stages of a disease, like in NAFLD or NASH, NASH. We can still use TradeSeq there, right, to understand trajectories for specific cell types. So I think this comes back a bit to one of the first questions is, when do we have a trajectory and when do you not have a trajectory? Again, a lot of it, I think, depends on the biology. If you have different stages of those diseases you mentioned, and you expect you'll be able to capture them with a decent resolution, where you go from one state to another state, and there's some kind of dynamic switch or dynamic development between them, then it could be possible to look at trajectories, but a lot of this is pragmatic. Hector, Kelly, anything?
Yeah, I think usually, we talk about pseudo time as if it's a time axis, but this works for any cell type or collection of cell types that exist along a continuum. It doesn't have to be a developmental lineage.
We have one new question. Sometimes getting the knot right in the position of the data, for example, just before divergence of a lineage, is difficult. Could the median of the slingshot clusters be used as the position of the knot? So by default, we separate out knots according to quantiles. Because if you have a lot of data, you might want to get some knots there to pick up the patterns that are maybe shown by those data. This is indirectly related, I guess, to medians of clusters, because if you have a lot of cells, you might get a lot more clusters. I think that's all the questions. So
just as a follow-up on that, for now on TradeSeq, we do not allow users to specify where they want the knots to be, just the number of knots.
Okay, thanks everyone. Enjoy the rest of the conference, the last day.